Sir, we
your
are live, sir.
అనిత ఒకసారి సార్ స్క్రీన్
No,
టెస్ట్
no,
చేస్తున్నారు చూడు. ఇట్స్ నాట్ విజిబుల్ ఎట్.
Okay, I'll do it once again.
జస్ట్ షేర్ కొడితే కనుక అది స్క్రీన్ షేర్ అయిపోతుంది.
Yeah. I used to it, but then I'm used to Zoom, Teams, and this is something new. Can you see it now?
No.
Because it's showing that StreamYard.com is sharing your screen. Stop sharing or hide. Shall I do one thing? Maybe there was an
అంటే ఇంకా
option.
స్క్రీన్ షేరింగ్ ఏం పర్మిషన్ అక్కర్లేదు కదా.
Sir, you are sharing your screen, sir. You have to share a window.
Yeah. I have shared my window.
Yeah, share entire screen or window. In that
Yeah.
Sir, Raveendra, sir, you have to share your.
Yeah, wait.
You have to share your window, not your screen. Sir, Raveendra, sir, you have to share your window, not your screen.
Just a minute then. See, let me explain you how it is showing for me. When I open share, there is an option for slides, video file, and share screen. Okay.
Sir, share screen.
Share screen, yeah.
Dialog box will be open and you will be having 3 tabs: your screen, window, and some other thing will be there. So on that you click window, and when you open your PPT,
is it fine now? Purna? Now
Yeah,
I
just
am
wait,
getting...
sir. Just wait, buffering and buffering.
మేడం మీరు నెట్ కనెక్ట్ చేశారా? వైఫై ఆఫ్ చేసేస్తాను. Madam, it requires my permission, madam. కనెక్ట్ చేయాలి. సిస్టమ్ ఫోన్ రెండు వైఫై కనెక్ట్ చేయొచ్చు. Sir, am I audible to you, sir?
Yeah, you are audible, ma'am.
You just stop sharing.
Okay.
Again, start. You just share your screen. Start sharing. So on that you'll have slides, extra camera, video file, and share screen. You just click share screen. So it will be
Can you see me now? My screen? Not yet. Can you see, ma'am?
Sir, am I audible? You are unmute, sir.
Yeah, you are audible,
Unmute,
sir.
sir.
You are audible.
Yeah. Yes, sir.
Can you able to see my screen?
You,
Sir,
not
you are
sir,
sharing the
visible.
screen, not the window. You have to share the screen, na?
Yeah, ma'am, I am doing that. See, there is three options. One is entire screen, window, Chrome tab. Which one you want me to share?
Window.
Usually it should be entire
You have to press
screen, ma'am.
window,
So,
sir.
window. Okay.
When you press window,
Can you see my
no.
screen?
Entire screen means your PC will be shared.
Yeah, see now. Yeah, I
Ah,
understand. Can you
yes.
see my screen
Now
now?
I am. No, sir.
Not
No,
yet.
no. You just enlarge it.
Yeah.
You go for slideshow for that show. No.
No.
You just go back. You put a slideshow, press escape, and share the open slide. Not this one. If you put this one, when you go in the slideshow mode, it will not be shared again.
Okay. You mean to say just guide me.
You just go back. You just go back again.
Yeah, stop sharing. Okay. Just
You
I'm
just
going to.
stop share it.
Yeah.
Stop sharing.
Yes.
Again, you go for share. You just go to the PPT. You just go to the PPT.
PPT means yeah.
You just go into the presentation. Your presentation. Just go to the PPT and open presentation mode
Okay.
and press Alt tab so that your Chrome window will be open. Alt
Yes.
tab.
Alt tab. Yeah. Chrome window. Yeah.
Your Chrome will be open now.
Yeah.
So now you start share it, share option, share option, and in the share option you select, go to the window.
Yeah.
Yes. Yeah, it's visible, sir, now.
It's visible, right?
Yes.
Yes,
Yes, it
sir.
is visible.
So shall we start?
Madam, please start the session.
Yeah.
We
Yeah.
three will go
Only.
out of the background. Madam, you just ask the session.
Okay. Very good afternoon, one and all. Welcome to the international webinar on formulation development of generic products, organized by Raghu College of Pharmacy, Vishakhapatnam. And today we have with us a speaker, a very good speaker and a scientist, as well as a good friend of mine from M Pharmacy Bits Pilani, Mr. Raveendra Nagella, who is working as a senior manager, formulations R&D, Hikma Pharmaceuticals, Jordan. So hearty welcome you, Raveendra, and thank you very much for readily accepting our invitation also. Thank you so much. So at the outset, I would like to invite the chairman, Raghu Group of Institutions, Shri K. Raghu sir, and our principal and convener of today's workshop, for this international webinar, Dr. Jagdish Panda sir, myself, Dr. Purna Nagashri, the co-convener and professor of Raghu College of Pharmacy, Mrs. Anita Raj Panigrahi, the event coordinator, dear faculty members, students, and all the participants. Hearty welcome to you one and all on this international webinar conducted by Raghu College of Pharmacy. So now may I request Dr. Jagdish Panda sir, Principal and Professor of Raghu College of Pharmacy, to give his introductory remarks. Over to you, sir.
Thank you, Dr. Nagapurna. And it's a great indeed, it's a wonderful day today to being with us. Mr. Narendra Babu, the senior of formulation and development, and from the Jordan. And thank you very much, sir, for readily accepting our invitation. And and definitely today is the Delhi. All the participants will have a very good extensive lecture on the formulation development, and definitely he is as a senior manager in R&D and in various pharmaceutical industries in India and abroad. Right now, Mr. Raveendra is working in Hikma Pharmaceuticals, Amman, Jordan. In spite of his lot of work pressure, and today he has with us, we have with him, we have Mr. Raveendra to give an extensive lecture, and definitely this lecture will help the every researcher in the academic line, in the students who are pursuing the research, and who are the participants, budding pharmacy students in B.Pharm and M.Pharmacy. Definitely it will highlight the almost every corner of the development of the formulations. And about this college is under the Benevolent Management Educational Society, so which is established for the year. I have started this pharmaceutical technology and pharmaceutical. And once again, I welcome all the participants and colleagues and my own students of this college and various colleges. And definitely, you will have the very good ample of experience, and there will be very good, discussion and question and analysis section will be at the end, and you can have a very good clearing session. And hope this will have a memorable experience in this webinar. And thank you, Madam Purna.
Thank you very much, sir. Now may I invite Mrs. Anita Raj Panigrahi to welcome the speaker with his brief resume. Over to you, Anita Raj.
Thank you, madam. Good afternoon, respected chairman, sir, respected principal, sir, senior professor Purna, madam, and my dear colleagues. I welcome today's the chief guest of our international webinar, Nagella Raveendra Babu. Having 17 years of vast experience in formulation and research, Raveendra Babu sir has completed in B.Pharm in the year 2002 with his post-graduation by the year 2005 and PGD PL from Nalsar University. So, sir has been associated with various reputed industries like in Alchem Pharmaceuticals as a research associate for the year 2006 and 2007, as a junior manager with Dr. Reddy's for the same period 2007, and as an assistant manager in Milan Pharmaceuticals for the period 2007 to 2012, and ABDI Ibrahim as a formulation specialist for the duration of 2012 to 2014, and with the Teva Pharmaceuticals as a principal scientist from 2014 to 2021. And currently, he is working as a senior manager from Hikma Pharmaceuticals since 2021. And it my great pleasure to invite such an eminent person for this international conference. Uh, Sir, I welcome you for your presentation for this webinar. Over to you, sir.
Yeah. Thank you, Anita, and thank you, Dr. Jagdish and Purna, for your kind words. Just you all can see my screen, right? Purna, can
No.
you see my screen?
No, no, Ravindra.
You
You
can't
are
see it.
visible now.
I am only visible.
Yeah, yeah, yes. Oh no, this is no, we can't see your screen. 10 minutes back we
Went
saw
back.
your screen.
You share again, sir, please. It went back.
Okay. Now you can see it, right? Purna, can you see my screen?
Ravindra. Yes. Yes. Good.
Okay. Okay. See, thank you for your kind words. Since you have explained me, I apologize for here I said in Alchem from 2006 to 2007. It is from 2005 to 2006. Okay. uh, just to let me start with my expertise. Most of my expertise in solid oral dosage forms like tablets, capsules, and I worked on different types of tablets like immediate release, delayed release, sustained release, and gastroretentive dosage forms, powder for oral solution, and suspension. In starting of my career I also did for powder for injections. So this is my expert area. Just to explain one thing, I was attended a deposition in New York in USA for one of the patent lawsuit by the brand company. I will explain you what is this lawsuit and why this happens, maybe in my subsequent slides, so that you will get connected to what I mean by deposition. Okay. Further, if you ask me about the expert areas, I have been into the formulation research. As a part of formulation research, you should be you should know about bioequivalence studies, intellectual property rights, and QBD, and design of experiments. These are my expert areas. Okay, just before I start, just I wanted to check with Purna. Purna, I'm audible, right? Anita or Purna, can anyone
Sir, you are
confirm me
audible,
that
sir.
I'm
You can continue.
continue, right? Okay, fine.
You are audible, audible.
Okay, thank you. Thanks for confirming. See, though, as an agenda, we have kept a topic like formulation development of generic products. But my efforts that after this one and a half hour seminar, uh, I will try at least, most of you would be like B Farm or M Farm graduates. or postgraduates. So end of my seminar, I will try to ensure that you would be clear what all the things that happens in the industry. So end of the seminar, you should be clear, like how you have to build your career path. So that is the big picture of my presentation. Rather than the in-depth knowledge of this, I try to ensure that you would be aware of the things which happens in the industry, so that you will further nurture the things which I discuss in this presentation. Okay. I will start my presentation with a simple story, and then eventually we will move in depth into the development part. Okay. See, if you look at this slide, necessity is the mother of invention. This adage goes, how old this saying is. This was by Plato. It was said in BC 400. I mean, currently it is 2400-year old saying. This was true, this is true, and this is going to be true in future. There are certain statements which will never change. When I say this, there is a reason. Like, a million years ago, when human beings discovered the fire, that was the necessity. And if you remember, I'll try to give some contemporary examples so that you will connect better. Maybe 2-3 months back, China tested supersonic missiles. That was the necessity. Why they tested? They believe there would be enemies and they want to protect. So that was the necessity. So that's why they did some research. And even better contemporary example is coronavirus. So I needless to explain much about coronavirus, because what the discoveries happened during this period, like it can very much relate to these things. Like the testing kits was a research. Okay. And then what were the vaccines which were invented? Those are all again a necessity at that period. If you look at the treatment regimes, the new drug products which were discovered during this phase, again it was the necessity for the humankind. So these are all the necessity. Why we do inventions is because it demands necessity. Necessity is the ingredient which spurs the research. So even if you look at the medium, maybe 2-3 years back, until 2-3 years back, I mean, before coronavirus, we are hardly I remember any time we are interacting through Zoom or MS Teams, or for that sake, the StreamYard, which is a new. We never used to interact through these mediums, but then because of the coronavirus, we all started, and industries like software industries has evolved to create a medium through which you can sit in a home, or and you, with a personal PC, you can interact. Again, this was the necessity of our. That's why the research was done in this way. Because being in a pharmaceutical industry, we used to connect through telecons. But though we had mobiles, we used to interact with video conferences. But then, like, interacting through Zoom, having thousands of participants all over the world who can communicate each other, this way of research was happened very recently. Again, see, because that's again the need. So the need is driving us to do research. Again, just before I go, research can be, just I can define research into two ways. One is driven by individual, and sometimes some people will take research as a profession. So there are differences between when an individual does research because of the passion. So there, usually when we drive with a passion, there won't be much timelines, and you do research by your passion. But when you take research as a profession, so like it's not like a group of researchers gathered together uh, to achieve a common goal. Now, I will explain how things will happen in my subsequent slides. How we will define a common goal when group of researchers gather together. Like it is very easy. If I am passionate towards a robotics, I'll do research in robotics. My goal is simple. But as a corporate company, when they do research, how they define their research goal, that we need to learn. Okay, just I will start a short story of a new molecule. Rather, I don't say it as a new molecule, because this tolvaptan is a molecule which was which was approved in 2009. Just it is a very good story to explain you. That's the reason I have taken this as an example. Being being you guys are pharmaceutical professionals, you know what is diuretics, right? Which increases the productivity of urine. Like it includes the diuresis, a chemical substance that increases the urine production. Now, the how we do research, like let's suppose, how we create the targets. Let me explain with this tolvaptan story. So, if you have, you might have read, gone through the diuretics as a chapter in your chemistry, in your pharmacology, and you would be very much aware when you give these diuretics, they have side effects. Okay, you along with water, you will lose electrolytes as well. So always you read or you by heart the side effects, like hypernatremia or hyponatremia or hypokalemia. All these are the side effects. So one of the marketing representative, this molecule was by Otsuka Pharma, Japan company. Okay, so when one of the field officer, like marketing representative, visited the doctor, so the doctor had put forward a question to this medical rep, like, don't you have a product which can just excrete water but not electrolytes? In fact, that question has triggered. Eventually, this representative carry forward this question to the management, and this has spurred further invention of this molecule, like tolvaptan. Unlike other diuretics, it excretes only water. That is the beauty of this molecule. Okay, so again, here, doctor, rather than the doctor, the customer. Who is the customer here? Patient. So patient gets the feedbacks. Doctor gets the feedback from the patients, and then this feedback is carry forwarded to the representative, and then this is carry forward to the management, and then that way the research happens. That is one of the reason, like, how we will identify the targets. Then eventually, so this product was launched in 2009 as a diuretic. But then how, again, there is another product, this same molecule was also approved in 2014 or 15, I believe, for another indication. Like the indication is ADPKD, which means autosomal dominant polycystic kidney disorder. What is this autosomal dominant polycystic kidney disorder? See, in this disorder, if you can see the screen, like, the kidney, where there are small cysts, like, small sacs which contain accumulated of fluids. So eventually it will get enlarged, and the patient go for end stage renal failure. Okay, it is a genetic disorder. Like, hardly, like, when I say a genetic disorder, only very few population suffers with this disorder, and there was not much focus on this disorder. Like, when you have one person in 100,000 population, then nobody focus, because all this drug research done by the corporate companies. So though there is always there is a motive is, you will, if you have a lot of demand for the product, you will get the return, because a drug research is a costly affair. When a new chemical entity is synthesized, as per the these things, 2.5 to 2 point, there was somewhere some 2.5 billion US dollars should be spent for a new molecule. So someone at American University evaluated this tolvaptan for this ADPKD, and they have published in a journal. Eventually, then even then, Otsuka Pharma was not interested in this. Okay, there was a journal. Eventually, one of the employee in this Otsuka Pharma lost the life because of this rare genetical disorder. So this spurs that management that, this is a rare disorder, we need to find out the solution, because again, whenever they want to go for a additional indication, again they have to spend money. So based on that research article, they spent some money to understand whether this molecule can is also works in autosomal dominant polycystic kidney disorder. And ultimately they did clinical trials, and they found that this molecule is also effective for this ADPKD. And then they got approval in US for this ADPKD. So again, how based on the need we do research. Let's go to another an example. This are most of you would be very much familiar about this gastroesophageal reflex disorders. Like, many of you at one point of time might have used this Dizin or Gelusil, or So how again, this is again why I'm explaining this example, to understand how a research would be done. Let's suppose, if you ask me, what does this Dizin contains? It is aluminum hydroxide and magnesium hydroxide. What does ENO contains? It is a sodium bicarbonate, sodium carbonate, and citric acid. Okay, so usually when maybe 40-50 years back, when people were suffering with gastritis or acidity, the easiest solution was like, because acidity is because of the acid, use something a base which can neutralize and which can subside. Okay, so people started using this Dizin and Gelusil, and even for that matter, even these medicines are available even now. But then the point is, you cannot these things are symptomatic relief. They are not giving any like a permanent solution for that. So again, there was a need how you need to control this. Like, if there is an acidity, you use it will give a relief for 10-15 minutes. After that, again your problem will continue. Dyspepsia, gastritis will continue. So there was a need. So people found the targets, like they understand that histamine receptors was identified. And they scientists know that, this histamine bind to H2 receptors in stomach. Then eventually it will trigger the production of acid. So then people started working on how we can stop binding of histamine to histamine receptors. So eventually the drugs like ranitidine, cimetidine, which are H2 receptor blockers, was discovered, and that they were prevalent in the market for pretty long time. But what is the disadvantage of this H2 receptor blockers? These H2 receptors, like acetylcholine, histamine, are the major chemical messengers in the body. These are prevalent throughout the body. These H2 receptors not only present in stomach, but in the cardiovascular system and your brain, your pulmonary, in the pulmonary system. Everywhere these H2 receptors are present. But what is the disadvantage of this ranitidine? When you administer ranitidine to to get relief from the acidity, eventually it will block the H2 receptors which are present throughout the body. So it has started giving its side effects. And moreover, you cannot use this ranitidine, cimetidine for more than 2 weeks therapy, because eventually it will give a lot of uh, permanent problems. So there was a need again, like, this acidity is not a temporary, I mean, it's not 2 3 days. Sometimes people have to use it for years. So then there was a need. So someone, like, then people, there was a discovery of a proton pump. Then people realized it's a proton pump which is there in a gastric mucosa, in the cells, mucosal cells. That is a trigger that causes the release of acid. So then people started working on how we can block this proton pump in the gastric mucosa. So the multinational companies like AstraZeneca, Takeda started doing this research from in 1970s. So eventually in 1979, omeprazole was launched by AstraZeneca, and that was a pretty famous drug. And eventually then there was like multiple proton pump inhibitors came into the market. Omeprazole, then it was a racemic mixture, like R and S form was both mixed and they used to give it. Then the potent form was, then people eventually identified that both R and S forms are not important for therapeutic activity. It is the S form of omeprazole is active. So eventually that has paved a road for esomeprazole launch. When I say esomeprazole, esomeprazole was launched into the market. Then there was rabiprazole, lansoprazole. So whole lot of the proton pumps were there in the market a pretty long time. Even today, most of the if you go to any doctor, I don't see anyone, like, who doesn't have used this proton pump inhibitors. But if you recollect these things, how you use these proton pumps, you have to take early morning before breakfast. Okay, what is the reason why you have to use it early morning before breakfast? Reason behind these proton pump inhibitors are get degraded in acid media. So what you need, so formulation scientists, what they did, omeprazole, they have done entry coating because it degrades in the acid medium before absorption. So you have to entry coat this proton pump inhibitors. So all proton pump inhibitors, or most of the proton pump inhibitors, are entry coated. So uh, to get an action, there would be a delay. Like, any entry coated product start acting after 1.5 2 hours, because it has to pass through J track, and then when pH starts after 5.5, then the entry coat will dissolve, and eventually there would be absorption of these proton pump inhibitors from the bloodstream. Then they get transferred into the parietal cells of stomach, and then they will stop. So there would be a delay of action. So usually, people who suffer with headache or acidity, people look for immediate relief. So then, if you look at, there is a gap, because you can't get immediate solution for this use, even through this proton pump inhibitors. Then there is there is a lot need to done. So again, eventually, if you look at the current trend, latest research in this area is how we can block this is potassium pump inhibitors. How does this potassium pump? First one is proton pump inhibitors. Now current trend is potassium pump inhibitors. It is a very latest trend which I'm talking about. The advantage of this is, you need not, I mean, again, why potassium pump inhibitors? Because again, the target was defined, like, rather than uh, proton pump inhibitors. The reason how these proton pump inhibitors are activated, these are activated, the reason for activation of these proton pump inhibitors are potassium ions. So rather than stopping this proton pumps, why can't we stop potassium pump? Okay, this was the target which was identified. So now, based on this target, companies started working on this potassium pump inhibitors. Now, what is the advantage of this potassium pump inhibitors? Let's suppose potassium pump inhibitors, these are a quick action. See, these these are not yet approved in US now, but then these are approved in Japan, South Korea, and other Asian markets. Eventually the clinical trials are going on in US, Europe, and within a year or so these are going to create $1 billion revenue in the market. So these potassium pumps, so the advantage of potassium pumps is, considering being these are weak bases, it does not get, it won't get degrade in acid medium immediately, and you don't need to do any entry coating. So that's the reason you can have a quick action. So that is the advantage of this potassium pump inhibitors. If you look at Vonospire and other figures which I have given here, are these are potassium pump inhibitors. So this is how the research, how the necessity drives the invention. Now, this is about whatever I have discussed until now is about how new chemical entities discovery starts. Now, let's come to the generic market, generic formulation business. How we identify new projects. If you remember, I said research can be individual, or people can take it as a profession. When individual, you are clear about your goals. When you go to a farm, if you associate for a research in a company, pharmaceutical company, then you have to identify your research goals. The company has to identify research goals. How the research goals would be identified, let us understand. So if you would have seen in the 12 Upton story, usually marketing representative who interacts with the doctor or with the pharmacist in the medical shop gets the feedback, and based on their feedback, this would be forwarded to the business development team. See, again, you can be open, whatever the departments which I am talking, as a pharmaceutical graduates, when you come out of the college, you have the opportunities in these areas of the pharmaceutical industry. So then you can see it as a pharmacist, you can be after B Pharm, and then you can be a medical representative. So this takes the feedback and they will give it forward the requirements to the business development team. So what is business development team? Usually in pharmaceutical industry, they do a segment wise means there would be a team who promotes the cardiovascular systems, so that is a one segment. Then endocrine system, that is a segment. Anti-cancer, it is a segment. So people team works in a teams for a segment. So this representatives gives the need, like what is needed for the patients. Let's suppose if doctor says that in management of hypertension, we are giving diuretic, we are giving vasodilator, we are giving other amlodipine, the other inhibitors. Let's suppose we are giving 2-3 compounds to manage hypertension. Then because the patient has to take 3 tablets morning, evening, so it's a patient compliance problem, is a patient compliance. Then they do some work, like they pass on this message to the business development team, because they say that this is the need, like instead of 3 molecules separately giving, what if you combine this together and give it, can you do this for us? Okay, they don't worry too much about like the science, whether it is compatible, incompatible. So they give some wish list, like this is the wish from the patient, or this is the wish from the doctor. So they give, this is the wish list. So this wish list would be prepared by, with the information from these representatives, this wish list would be prepared by business development team. Okay, then this business development team, what happens is, then they analyze this, okay, this fine, this is the lead where we can do research. Then they will analyze, like they see uh, uh, they get the market intelligence. What do you mean by market intelligence? Who are the competitors, like what is the business in this segment, Okay, when I club these three together, maybe I have to do some other extra studies, I may have to do some investment. Okay, what would be my sales? So this, with this leads, they do some research again from the, based on the marketing intelligence. So how does this marketing intelligence happens? Usually these are consultants. One of the famous consultant is in India. There are many consultants. One of the famous consultants is eValueServe, where people have they pay a lot, like, again, if you come out of the college, you have opportunity to work in market intelligence. So what is the role of this market intelligence? Like, they what they do is they will prepare a market intelligent report of a particular drug. Let's suppose X drug is sold by Dr. Reddy's, is generating this much revenue. The same drug by marketed by Sun is selling so much. So like that, they will compile, and just they will, based on the data, they will provide this report. Then this business development team, what they do is, okay, so the, what is the potential for this particular molecule? Okay, so we are selling like X plus Y plus Z, so this amount we are selling. Let us suppose how many prescriptions are coming together, like X, like three molecules, how much prescriptions are coming. They will club and they will make one assessment. Let's suppose if I do some research by combining these three molecules together, I can sell this much amount. Okay, so they do, they put some assumptions, and they will give these assumptions, and uh, this would be forwarded to the portfolio management. Again, when I say portfolio management, as BD teams work segment wise, like the wish list from the business development team, from the cardiovascular, endocrine, anti-cancer, all different teams will provide their wish list to the portfolio management. Then this portfolio management, how does this portfolio management works? This person takes the wish list from the business development team, and then they will give this wish list to the formulators, and they will ask, like, this is the wish list, hi guys, can you check this, can you manufacture? Then you formulate them. Now as a formulators, we need to understand, like, this A plus B plus C are compatible or not compatible. Then so this wish list would be sent to not only to the formulators, it would be sent to formulators, analytical department, and intellectual property rights, and procurement team, and biopharmaceutical team. These are 4 5 departments. They will send this wish list. Can you work out, can you check whether it is feasible or not? Per se, for a formulator will check whether we can combine all this together, and then intellectual property rights members will check, is there any patents which will prevent us doing this, developing this product. And then procurement team checks whether is there availability of API and all are available or not. ARD will check whether do we have this required analytical instruments are available or not. Based on all these things, then we will we will give our budget. Let's suppose, so okay, fine, with the existing infrastructure what we have, we can take up this molecule, this project, but then this is the budget what we would be required. Then this all information again would be passed on to the portfolio management. Then this portfolio management team, what does they do? See, business development will give the assumptions, like, we can sell this molecule. If you give this project, if we can sell for next 5 years, let us suppose today we are in 2022. If you take up this project, if we launch this product in 2025, from 2025 to 2030, for 5 years, this would be the this much amount of tablets, we can capsules we can sell. This is the forecast that would be given by business development team. From the R&D, what we give, we will say that if you want to develop this product, this is the budget we need, this much of the API we need to procure. If we don't have, let's suppose we don't have some instruments, then we will give the capex. This is the budget, this would be the expense of this product. Then then portfolio management team takes the input from BD, formulators, and everyone, and they will compile, and then they will give it to the financial feasibility. Then we need to check it, right? These are not small, like, it's not in thousands. We have to spend the investment in in Indian rupees crores, 100,000, maybe more than crores. So then there would be financial analyst. Then they use some tools like what is the NPV, what is the compound annual growth for this product. Like no, they put, if we sell this much amount, if we spend this much budget, what would be the NPV? Usually NPV is the net present value. This is one of the financial tool to understand the project is feasible or not feasible. So based on NPV, then they will say that okay, this if you spend this much amount, if you sell this many tablets, and this is if then this would be the NPV, or this would be the compound annual growth rate. This information again would be passed over to from finance department to portfolio management. So then after all this exercise, this would be presented to the project selection committee. Who are project selection committee? Usually those are the board members of a company, or CEO of the company would be there. There they will present, this is the project, this is the proposal, this is the benefit, can we take up this? So the project selection committee is the group of decision makers. So they will think, okay, fine, this is the NPV, this is the CGAR, so we can take, or we don't want to go ahead. So they will take a decision. So this is the way how new products would be identified and would be taken up for the development. So you have, so once you complete your career, I mean once you complete your degrees, so these are all the options you can look forward in an industry. You, based on your skill set, you can choose based on your interest. This is in a generic. Apart from this, there is manufacturing, QC, then quality assurance. There are many other departments too. Now, let us understand what is difference between brand and generic. Usually, in India, in Indian context, every company sell their drug in brand names. But you need to understand from the other country perspectives. Brand means the person who has first launched the product into the market. That you call it as brand. Generic means subsequently whoever has launched the product subsequently after the brand, then they would be generic company. Let us suppose, just give you an example with a contemporary example of molnupiravir for COVID. This is one of the drug which was which got emergency use authorization in US. First it was got emergency use authorization in the Europe. Subsequently it was also got emergency use authorization in US as well. So who was the who got this first? It was invented by Merck. Though when I say Merck, this molecule per se was first discovered in university. Eventually the corporate companies buy this from the university, and then they did clinical trials, and then they have launched this product. So the first product who has launched molnupiravir was Merck. So Merck is selling this product under the brand name of Lagiviro. So now who is the brand company here? It is the Merck. But if you look at other companies also marketing this, like in India, you have Dr. Reddy's is also marketing, Sun, everyone is marketing. So those are all would be generic companies. Okay. Similarly, there is another medicine which was which was discovered by Pfizer, that is Nirmatrelvir. This is also it is also shown very much efficacy against the COVID. So this was marketed under the brand name of Paxlovid. So who is the brand company here? It is the Pfizer. Subsequently, whoever market this product, it would be generic company. Now, I want to drive together more towards to differentiate brand and generic. Why this concept? Because in Indian context, brand generic has no meaning, but the concept of brand and generic is from the regulated markets. In subsequent slides, I will try to give you more clarity why brand and generic and what benefits it gets, what benefit the patient gets. Okay, let's suppose take an example of pomalidomide. The brand company is Celgene Corporation, and brand name is Pomalist. It was approved in US in 2013. If you look at this capsule cost 21 capsules cost 21K US dollars. So in Indian rupees, each capsule cost, based on dollar value today, each capsules cost 75,000 rupees. Okay, this is an anti-cancer medicament, and it has to be taken at least 3-4 months. So people, I mean, if the brand company want to sell in India, like, you have to take a 21 capsules, you have to sell your property, whole property. People will rather die, take rather than taking this product. But then the same capsules in India is available at the cost of 4,000 rupees. But then again, we will understand this in subsequent slide. So because 4,000 rupees. Okay, next come to the atorvastatin calcium. Again, this is one famous example. And clopidogrel, as a brand name of Plavix, was also by brand company Pfizer. So when they launch this product, when brand company launches this product, they sell at exorbitant price. This Pfizer, when launched Lipitor, the cost of the brand product was $5 per tablet. But when first generic company entered in the market, the price was slashed to 0.3 cents per tablets. So that's why generics are very much lucrative, but from the patient perspective. Now understand why brand companies sells at exorbitant price, and why, because to identify new molecule they have to spend money in billions, and that, they have to get back that amount they have spent in the research. So they will sell at exorbitant price to get back the money what they have spent. Now let us understand, again, this is from the context of US. When we talk about generic products, we are talking from the regulatory market perspective. How does the insurance works in USA? For in India, like, you can take any medical insurance, you pay premium, and when you hospitalized, then insurance company will pay you back, and the insurance company pay to the hospital on behalf of you. Like, there is no role of government here in India. But in case of US, it would not be in that way. Like, the medical insurance is sponsored by US government, because as per one study in 2017, US government has spent 705 billion US dollars in reimbursing the medical expenses. It's a very huge amount, because if you look at in my previous slide, you have seen pomalidomide, what is the price they are selling, brand companies are selling. When they launch, they will sell at exorbitant price. Again, who gives, I mean, who pays for this? It is the government pays. So that's why people can afford. But in India, people can't afford that price. So again, there are two types of funds which are directly managed by US Treasury. One is Hospital Insurance Trust Fund. How does this fund would be created? Like, taxes paid by employees, employers, and other income tax paid by paid on social security benefits, interest earned. These are all accumulated, and this would be used if a person hospitalized. At that time, from this fund, government pays to the hospitals. There is another fund called Supplementary Medical Insurance Trust Fund. This is also managed by US government. This usually used for purchasing of the outpatient drugs. Now there is a reason why I'm explaining these things. Okay, now excuse me, give me just a minute. So as I said, because the government to start a brand product, as I said, to launch a new product, it requires around 2.5 billion US dollars a minimum. So they need to do a lot of research to launch that product. So like they have to do preclinical trials, and it is not that you discover something in the lab, and if it has shown effective for one indication, then you just can't launch. You have to explain what is the advantage of the existing molecules, is if the new discovered molecule has any advantage or more effectiveness, does it have less side effects. You have to give a proper justification. If you, I mean, just like that, it has a benefit, I mean, it has an effective and XYZ indication, FDA will not allow you to launch unless and until you show the superiority about over the existing molecules. You have to do that. Then you need to do animal testing, like you have to do toxicity studies on animals, mice, whether what would be the impact when we administer this in a long term, like does it can produce a cancer. So these all things to be conducted at preclinical stage itself. Then once this is done, then it will eventually move on to the phase 1 studies. So phase 1 studies from animals to humans, first time in humans. You administer, you don't know the dose, how much dose I need to administer. Again, they have to do those escalation studies to identify what would be the optimum dose that can be given to the humans. This would be done during phase 1 study trials. Usually this is conducted around 20 to 80 healthy volunteers. This would be done to understand what would be the minimum, I mean, to understand what would be the maximum tolerable dose in the humans. Once it is done, then it will go for the phase 2 studies, where it would be conducted on the patients. Around 150 to 300 patients, 150 to 350 patients would be used to understand the safety, toxicity in the patients. Then in phase 3, then the number of volunteers will increase. Then there in phase 3, they will find the efficacy studies. Let's suppose if I say in one of my previous slides, I said potassium pump inhibitors. So I found this, but then you have to prove what is the advantage of potassium pump inhibitors against proton pump inhibitors. So you have to show your superiority. When you say that effective, it is fast acting, then you have to demonstrate that. And if you have any side effects, you can give that justification as well. If the existing product has a side effects, if I say that the new molecule has no side effects, then that is also a good justification. So you should have a justification and superiority over the existing. Once you do all these things, then you will go for regulatory approval, and based on the assessment by the regulatory authority, the drug can be approved. Okay, based on the agency approval, you can launch the product in the market. After you launch, since during the clinical trials, you just the drug was evaluated around 5,000 patients maximum. In case even after launch of the product, you have to do we call it as phase 4 studies, or pharmacovigilance. Like it will go for I mean, vast number of patients. You need to understand, to record more adverse effects, side effects, etc. So this is called post marketing surveillance, When I say even once you come out of the industry, there is pharmacovigilance is one of the department where you have a lot of options. So this is the way how a brand product approval takes place. It is not like it is not a year or 2 years process. It may range from 10 to 15 years to launch a product once they identify their efficacy in the lab or in vitro. So it is 10 to 15 years process to get the approval of the brand product. Now understand what is the generic, how the generic drug approval process takes place. So FDA and health authorities believes that since you have done already the clinical trials like animal studies, phase 1, phase 2, phase 3 clinical trials, so they believe that, again, it is a costly affair. Not that, if somebody has done that, you need not to do all this, generics need not to do all this amount of investment on this. So the health authorities will rely on the data which was generated by the brand company. So generics need not to conduct all these trials. What they will do directly, they will go for the manufacturing of the tablets, and they conduct bioequivalence study, like, like rather than doing safety efficacy trials, they will conduct bioequivalence studies in a healthy human subjects, and they will match uh, plasma profile, plasma drug concentration of the brand product versus generic product. Okay, there are certain rules, how much close or how much you can deviate from the brand product. So you have to compare the plasma profiles of brand and generic. If you pass, and based on that, regulatory authorities can accept you as a generic product by relying on these all, which, I mean, preclinical clinical data on the brand product. So generics, the investment of generic company would be very less compared to the brand. That's why if a brand company sells the product at $5, then generic company when come into the market, they can sell at $0.3. So that is the that is the difference between the brand and generic. Now you can say government can impose to control the cost, but the problem is, like in India, there is, there is an act which regulates the price of medicines, like they call drug price regulatory act, DTPC or something. So based on that, the government regulate the price of the medicine. But then what happens if they start regulating the price of the medicine? If government decides that, nobody will do the, why corporate companies will spend billions of dollars and bring the new molecules into the market. So that's why in US and Europe, government will not interfere in the price of the brand products. Okay, so the price would be by the companies can decide based on the demand and based on the expenses they have spent during the research. So this is why, this is the difference between the brand and generics. Now again, how generic, let's suppose for in case of brand products, it will take somewhere around 10 to 15 years time to bring any new chemical entity into the market, whereas brand products, I mean generic companies, can bring the molecule into the market after the brand launch within 5 to 6 years. So the time and investment by generic companies would be less comparative to the brand product. Now let us understand some exclusivity rights that are there. Again, it is very much important to understand what are the different types of exclusivity rights. One, as you many of you know, like patents. Patents has, I can say, patents are negative rights. Okay, if anyone has any invention, anyone can file a patent and they can get a patent approval. But then again, remember, these are the negative rights. Negative right means, uh, if I got an approval, it doesn't mean that I am approved to market that. They say that you are the owner of this invention, and you can prevent others using your invention and use selling your invention. Why is so? Like, because try to understand in this way. uh, patents Patent is an authority where they are not experts to say that the particular one is like, uh, let's suppose, let us talk in context of medicines. They are not authorized to give an approval to sell a medicine. Let us suppose if you say that, now today I found a proton pump inhibitor. This X molecule inhibits the proton pump, and this will result in a decrease in the acid production. So this can be useful in gastritis. This is the invention. If you go, if they say that no one has ever, I mean, this chemical moiety, no one has applied for patent in around the world, and irrespective whether it is toxic or non-toxic, whether it is carcinogenic or non-carcinogenic, they will say that there is an invention. Nobody has claimed this particular compound effective against gastritis using potassium pump inhibitor. They will approve it. So they are not authorized. They don't do full scrutiny. But then if someone else uses same chemical entity, and if they come into the market, you have a right to sue them in the court and you can stop them. But you you get it, these patents can be used as a negative rights to prevent others to enter into the market, but it doesn't authorize you to enter into the market. Now there is a separate body like health agency. Like in every country we have, like in the US it is US FDA. In case of Europe, like I mean, European Medical Agency. And in case of Australia, this is again Australian Health Medical Agency, TGA we call. Then in Canada it is Canada Health Science. So there's a different medical health agencies that approve drug product approval. Okay, now let us understand what is data exclusivity. Basically, I'll explain this by comparing US and Europe. Okay, now US government has done a balancing act between, like, brands and generics. Reason behind brand company, because unless and until they get, if they spend 2.5 billions, if they don't get a profit, nobody do research for charity. So if you that's why I mean, US government has left the fixing of the price by the companies. So here, but then if you understand who pays this medical expenses in US, it is the US government. If you have seen in my previous slides, in 2017 they have spent 705 billion US dollars. So that now, if company, I mean, government want to bring balance between brands and generics, because uh, it's always like, it's a burden, right? If you every year you are paying, so they have to encourage generics as well. At the same time, in order to motivate the brand product, they have to go in a new research, so they have to motivate brand. At the same time, in order to reduce the burden, they have to encourage the generics as well. So what they did is, they have, I mean, they there is an exclusivity rights. Let's suppose anyone in the US first launches new chemical entity, they will give 5 years exclusivity. What does that mean? Like 5 years, nobody would be allowed to enter or market the similar product in the market. Let us suppose if I say molnupiravir in US, other than Merck, nobody else would be allowed to market that product. But this is a different story in India. Okay, so why this is data exclusivity for 5 years is given? So whatever they have spent during the research, billions of dollars, what they have spent during this 5 years, they will get a benefit. That's a monopoly, so they can fix their price and they can earn what they have spent now. But then it cannot be allowed for many years, because This is the data exclusivity given by US FDA. FDA will not allow any generic product filing until 4 years, because it is a 5 years. So US FDA allows a generic product file of end of the 4 years, because again approval process will take 2-3 years. So by the time uh, generics comes into market, it is 6 to 7 years. So there is some act called Hatch-Waxman Act in 1984 passed by US government to encourage the generics, because now they have given this flexibility for brand products to earn amount what they have spent. But then it is, people will become greedy. Like after 5 years also, what brand companies does, like they don't want to lose their profits, because the generics comes, they have to slash even their price. Because if there are two products are available with the same price, with same efficacy, which was approved by health authority, then why I should spend $5 if something is coming in 50 paisa or 50, I mean 0.3 cents. So people will go for this one. At that time, brand product lose their property, lose their revenue. So what they do is, apart from this data exclusivity, they do have some patents. They file some patents based with that negative rights. They will stop the generic entry. So it was becoming more and more burden for the US government. So they have brought Hatch-Waxman Act in 1984. What they said is, okay, fine, if any generic company whoever the first launches uh, uh, generic product in the market, and if brand company sues, like because of the patents, then generic company has to fight in the court. These court cases in US are very costly affair. So generic companies has to spend some amount, because but then, so what is the benefit if you fight and if you launch your product? Then based on Hatch-Waxman Act, you will get 180 days exclusivity. Means generic company will get 180 days exclusivity. What does again it mean? You would be there in the market, the along with the brand company, you would be alone there. So, I mean, the first company will remain 180 days. 180 days means 6 months. You can share profits. Let's suppose, usually any molecule, I say atorvastatin or clopidogrel, which I discussed in earlier slides, each product has more than $1 billion sale in 1 year. More than $1 billion sales. If such molecules are there, even a generic company along with the brand company there for 6 months time, even if you capture 1% of sale, you learn a lot, you earn a lot. So that is the benefit. The benefit comes at the efforts. You are challenging the brand company, you are invalidating the patent of the brand, or you are designing around the patent which is already filed by the brand. So you are doing an invention. You are either you can invalidate, or you do design around the existing patent of the brand. You challenge them in the court, and if you can file, you have a benefit. You can be there in the market for 6 months along with the brand product in this, I mean, so you are the two players which are there. So you can, whatever you have spent, you can learn, you can earn, sorry. So that is the benefit of generic companies based on Hatch-Waxman Act. Now, along with NCE exclusivity, which I said 5 years, let us, this is for new chemical entity. If give me an example, like if I launch a new chemical company, let us suppose tegoprazan, which is a potassium pump inhibitor. If I first, this is a new chemical entity, first time, if if it is approved in US, you will have 5 years exclusivity. Okay, let us suppose, instead of new chemical entity, if I say new clinical investigation, in case of tolvaptan, when I said they have given, there are two indications for tolvaptan. One is diuretic, another is it is also indicated ADPKD. Okay, if you do, because you have to do some other work on clinical studies. Like if it is a new investigation, then you will get additional benefit of 3 years. Even for that matter, let us suppose I have IR product. It has to be taken 3 times a day. Instead of IR tablet, if I design a sustained release formulation. You can take only one product a day. Again, you have to do efficacy studies. Some clinical studies you need to perform to show the efficacy. So again, you need to do not that the way brand companies do, but you need to do some more efforts. So for that, again, if you get your dosage form change, you will get 3 years benefit. Okay, if it is an orphan drug, what does it mean by you will get 7 years exclusivity if it is an orphan drug? What is orphan drugs? Because these are the rare disorders. In US, if the patient population is less than 200,000, if the patient population in US is less than 200 population is particular for a particular disease, then you consider it as an orphan drug, because orphan drugs means pe- only few people get affected. In order to promote, like diabetics is very well known, so there are a lot of people, so a lot of people will buy the medicines, so brand companies will get their returns immediately. But this, in case of orphan drugs, you don't get so much of benefits immediately. So for orphan cat, they had defined this orphan drugs, and if it is orphan indication, then your data exclusivity increases from 5 to 7 years. Again, this is the way how regulators or government encourages the corporate companies to do research on the unmet needs of the patient. Let us suppose tuberculosis, per se it is not so much prevalent across the globe today, but then it is very lethal. So because I'm not going to get the benefit of this if no corporate company works on research, then it will become a major threat to the population. So that's why they give some incentives to incentives or encouragement to work on the unmet needs of the patients. Okay, apart from this, let us suppose any new chemical entity, when it is approved, it would be for the adult population. Eventually, again, they have to do some safety and efficacy studies on the pediatric population. Even if you have seen, like in case of vaccine, when approved for COVID, it was only for adults population first given, then eventually it was given it to the children's. So when you do additional studies on the existing new chemical entities, you will get a 6 months exclusivity for pediatric formulations. Okay, this is the US. In case of Europe, in Europe, the data exclusivity time is something a bit changes. So there they will go for 8+2+1 regime. They say that, what does it mean? New chemical entity can be given 8 years data exclusivity, and that 2 years for they say that it is SPC grant, then there is 1+1 year additional regime you can get it. So that is in orphan drugs, again you can get 10 years. Pediatric exclusivity would be 6 months. Now let's come to the our main topic of generics. So in regulatory assumption, what is the regulatory assumption of generic products? They say that pharmaceutical equivalence plus bioequivalence, which is equivalent to therapeutic equivalence. So what does this mean? Let us suppose you are pharmaceutically equivalent. What do you mean by pharmaceutical equivalent? If the drug product has identical dosage form, if my brand product is a tablet dosage form, I am also tablet dosage form, and my brand product route of administration is oral, I am also, a generic company is formulating, it has to be through the oral route, and it contains identical amounts. Let us suppose if brand API is 10 milligrams, then brand should be 10 milligrams. Like I cannot say generic product. Let us suppose paracetamol. Take an example of paracetamol. If brand company is selling a paracetamol 500 milligrams, then being a generic, you have to sell only 500 milligrams. Let's say if you say instead of 500 milligrams, no, I want to sell 550, then you would not be a generic. You would not be allowed to call it as a generic. Again, you have to do additional clinical safety efficacy studies. So what is the what do you mean by pharmaceutical equivalence? It should be same identical dosage form, route of administration should be same, and it should be same molar concentrations or identical amounts of the active ingredients. When you have this, if you meet the pharmaceutical equivalent criteria, and if you establish bioequivalence means when you test your product and brand product in healthy human volunteers, if you compare your plasma profiles and if it is matching, then it would be deemed as therapeutic equivalent, like both can be used for the same indication, and you will get exemption from long expensive phase 3 studies. So because bioequivalence studies, compared to other safety efficacy trials and all those things, bioequivalence studies are very comparatively very cheap. Now, so let us understand what can be similarities or differences that can be allowed. So you should have same active ingredient, and you should have same pharmacokinetic and pharmacodynamic properties. You should have same stability and mechanism of action, safety, efficacy, route of administration, and indication. You see, again, let's suppose a brand is selling X product for let's suppose a brand product is selling for reducing the fever. You can't sell like, oh yeah, I manufactured this 500 milligrams. It can relieve the pains. No, you can't. You have to give the same indication of the brand. If you want to prove that it is additional indication than brand, then you have to do additional clinical studies, the way we discussed in the tolvaptan case. They have done additional clinical studies to gain ADPKD indication. Then what are allowed? Different salts are allowed, but then it changes from country to country. In case of US, what do you mean by different salts? Let us suppose clopidogrel bisulfate. Clopidogrel bisulfate is the salt. Okay, if brand company is using API as clopidogrel bisulfate, as a generic, you should use same clopidogrel bisulfate salt. But in Europe, it can change. In Europe allows you can use a different salt. Like if brand is clopidogrel bisulfate, you can use clopidogrel hydrobromide salt. So that is allowed in Europe, but not in the US. So then you can use different excipients than brand. If he is manufacturing the tablet using MCC, if you say that I want to use lactose, you are fine. You are free to use any excipients, colors, flavors, preservatives. This can be changed. Then you can have different shape and size and scoring. You can have different product expiration. Let's suppose if brand has given 30 months expiry, for the expiry date as 30 months, you can have 24 months or you can have 48 months based on the stability data what you provide. Then you can have different manufacturing process. He has manufactured using the wet granulation process. If you go for a direct DC process, it's fine. You can have different product name and packaging. You can have a different packaging, but I from the product name, because the brand sells as in the brand name, you generic companies sell by the generic name. Next, let us just understand this regulations changes from country to country. So if you understand, we can based on the you can divide across the globe countries into 3 baskets. One is developed countries, another is developing countries, third one is underdeveloping countries. Okay, though from the economy point of view, these are categorized based on the economy. Now in similar way, we can define in pharmaceutical industry defines the market into 3 categories. One is regulated markets, another is semi-regulated markets, third one is ROW. We call it as rest of the world. Example for the regulated markets: US, Europe, Canada, Japan, Australia, New Zealand. This would be deemed as regulated markets because they have a stringent regulatory requirements to get an approval to sell the product in particular countries. Whereas semi-regulated markets like Southeast MENA region will be considered as semi-regulated market, like Kingdom of Saudi Arabia, Jordan, Turkey, Brazil, and some other countries in this region would be considered as semi-regulated market. Rest of world, then India, Sri Lanka, Bangladesh, in many other North African countries would be considered as rest of the world. Don't directly correlate underdevelopment countries to rest of world. Just to make you to identify to explain the differences between the regulatory, semi-regulatory, rest of world. I have given example of developing and underdeveloping context. This is a context. So in similar way, based on the regulatory requirements prevailing in the particular country, we define them into three groups: regulated, semi-regulated, rest of world. Let me explain this more details, why I call it as rest of world, semi-regulatory, what are how the regulations define. So in India, if you want to sell, there are two authorities which give approval to sell a product in the market. You have Drug Control Authority and you have Drug Control General of India, DCGI. You have two routes by which you can get the approval. Usually, the DCA route, usually we follow, like Drug Control Authority can approve, provided provided a molecule is 4-year old in the Indian. Let's suppose first time someone has sold paracetamol in 2000. If another company want to sell that in 2005, considering 4-year old molecule in India, then instead of going to the DCGI, you just go down to the DCA, which is State Control Authority, and DCA can approve your product to sell in the market. But DCGI route you have to follow, provided if it is a new chemical entity and it is less than 4 years in the Indian market, then you have to go through the DCGI route. Just understand, because the requirement, what is needed in order to get a test license. Sorry, I could not open this PDF. It's almost only 10 to 15 point. Let me try to open this if I can. Okay. So what is requirement? Very simple. It's a one-page document. What is needed is just you need maybe it would be 10-15 page info which you need to give to get an approval in India. Not beyond that, nobody ask any questions about this thing. That's why we call the regulations are very less. What they will ask: where is your company, who are the promoters, and just what is the manufacturing plan of the manufacturing plan, and what would be like a schematic flow of the manufacturing process. Let's suppose I'm doing dispensing, granulation, drying, milling, tab compression, coating. This is one page. And then I say I'm mixing A plus B plus C, this and this thing, maybe 3-4 page document. You pay around 300 rupees, you will get an approval, and it will take 2 to 3 months maximum. And apart from this, you should have one manufacturing license if you are if a company is where you want to manufacture it. Okay, even that requirements is very less. So when you move on to the semi-regulated countries, what they ask, the requirement would be like you have to provide module 1, 2, 3, 4, 5. I will explain you what does it contains. And in regulated markets, same. Semi-regulated and regulated markets, same. But the amount of information which is required in module 1, 2, 3, maybe if it 3-4 pages document in these modules, that would be okay for semi-regulatory. But in case of regulated markets, they ask a lot of information. See here, if you look at number of GMP batches, like when I go for an approval in India and rest of the world, nobody will ask whether you have done a stability, do you have stability data, what test you are doing, whether it is meeting or not doing, have you done biostudy or not. If it is less than 4 years old, more than 4 years old, nobody will ask you. But in semi-regulated markets and regulated markets, you have to take a batch of minimum 100,000. Like it means You have to make 3 batches, each containing 1 lakh units, packaged units. You have to make 3 batches, and then you have to charge them in the stability based on the country requirement. You have to provide 6 months accelerated stability data, bioequivalence data, and other data. You have to file with the you have to file this with the respective regulatory authority. In case of KSA, you go to Saudi regulatory agency and you pay 20,000 riyals. Then it will take 2 years time to give the approval. Similarly, the case in the Jordan. In regulated markets in US, Europe, the amount of information that would be asked by the regulatory agency is enormous. And you understand, just you will understand if you look at the application fees to file a generic product in us you need to pay 197K, means almost 200,000 2 lakh dollars you have to pay as a fees to review your dossier. You understand? Let me calculate. $200,000 means in Indian rupees 1.5 crores. You have to pay as an application fees to review agency your dossier. Understand the important and regulatory requirement, and the requirement in this module 1, 2, 3, 4, 5 would be, again, you have to do huge investment. Even to launch a generic product in a US, it's not that easy. You have to spend a lot of money, and anything, it's like if you spend a lot of money, you'll get a lot of returns. Let us understand what is this module 1, 2, 3, 4, 5. So module 1 is you can go to the ICH guidelines to understand more about these modules. Just I'm giving a brief idea to understand the regulatory requirements between the markets. In module 1, it will be regional administrative information. The way I said, where is your company is located, who are the promoters, blah blah blah. This would be given in module 1. In module 2, it would be a summary of the things, like quality overall summary, then non-clinical overview, non-clinical summary, and clinical summary. This a summary would be given in module 2. In module 3, it would be quality module, where you give controls, CMC data, chemistry, manufacturing, and controls data, and non-clinical study reports in module 4, and clinical study reports in module 5. So these are the modules. Each module would be let's say a minimum 1,000 to 2,000 page document for regulated markets, more than that sometimes. Then in case of regulated markets, like as I said, in maybe compared to India, semi-regulatory and regulated. So regulated markets wants not only like for formulation details, they will ask you the API. Who is the API manufacturer? They want, say, let us understand what is drug master file. It is a submission file that is used to provide confidentiality information about facilities, manufacturing process, and articles used in the manufacturing, processing steps, packaging, storaging, and more. All these details, these are the confidentiality information. Like if you have this info, even you can start your manufacturing. This confidentiality information would be provided to the USFDA. These are the DMFs. These are called dossiers or DMFs. So there are 5 types of DMFs. Type 1 is manufacturing site, facilities, operating procedures, and personnel. Let us suppose this type 1 file is no more, but then since in order to prevent the confusion, they have kept even type 1 file, because now no more type 1 filing, because US FDA comes to your facility and they inspects and they give approval. Let's suppose if you say that I want to manufacture molnupiravir, then if you file your finished product dossier to them, they will after scrutinization, before giving approval, they will inspect your facility and they will see that you are following all quality requirements as per current CGMP practice, and then if they are convinced, then only your product would be approved, and then you can sell them in the market. Type 2 is active ingredient or drug substance intermediates files. Type 3 is packaging material. Type 4 is excipients, colorants, flavors. And type 5 is FDA accepted reference information. So they want, let us suppose, if you are manufacturing a capsule, they want to know from where you are getting API and how the API was synthesized, what were the key starting materials of the API, from where the API vendor has procured them, what were the controls of the key starting materials, what was the manufacturing controls in the synthesis of API, what are the impurities, what are the stability data, what is the MARF they are providing. So all this vast data, they need it. Not only that, they need even excipients which are going into the capsule. Who is the manufacturer, where they are producing, what is the quality, how he is controlling the quality. Similarly, each and everything which goes into the product, they want to know it. Unless and until they are satisfied with this information, your product will not be allowed. And once you file all this information, you are not authorized to change even the small step. Let's suppose first you are doing you are not authorized to change. Let us suppose if I am I'm procuring this microcrystalline cellulose from FMC. Tomorrow if I want to change from FMC to other supplier, I'm not authorized. You have to give this information to the FDA. FDA will evaluate and then they will approve it. So you have to provide all info to the FDA. But in semi-regulatory markets and ROW markets, nobody worries because you are not filing all this information. Let's understand how the formulation development happens. Usually, any other research, all things will start with a literature search. Then, after doing the literature search, in case of normal research, what we conduct in university, we rely more on the published literature through the journals. Though we do depend to some extent on the published literature, there are few government websites which, like the brand product, when they get approved, this would be available in these websites. Let us suppose in US FDA, once they approve, they upload all the info, all studies what the brand has done. That called summary basis of approval. On what study basis agency has given approval, that would be provided to the FDA, and FDA makes it available for all generic companies. So we usually refer and rely on this information. Like US FDA website, if you go, you will get this information. So in US FDA website, second one is Orange Book. What does Orange Book contains? Like, if there are any patents which are informed, like, when you file your new product to the FDA, you have to give the list of patents associated with that new molecule. So FDA will publishes those patents in the Orange Book. So when these listed patents, you need to understand, and you have to either you have to do design around, or you have to do the invalidate, and you have to provide a patent certification when you file your product. So this information would be available in the Orange Book. It is not that I need to do all patent search. You go to Orange Book, you will get the list of patents which brand is interested, and if you are out of those claims, and then you can go ahead and you can file your product. Then another is, third one is dissolution database. The third one is DAF. I need to open it to check it. The fourth one is dissolution database. Let us suppose brand company, what is the QC dissolution medium they are using, that would be listed. You can see, and you can follow same dissolution medium, and that those are about the FDA. Further, you can go to the medicines.org UK, and also EMA. You can refer to Canada. TGA is Australia. PMDA is Japanese. And apart from that, you can download patents from the ES SpaceNet across the world. So these are the majorly when we do go for a generic, we rely on these health authority websites to get information about the brand. Once we do all these things, we do patent strategy. The way I said, the brand company will file the associated patent uh, to that drug product. So we will evaluate those things, and we will see, like, what is the patent expiry, like when we want to launch it. If we want to launch it, we have to design around the patent. Or if the invention, you believe that it is invalidated because it is already available and the prior art, you can invalidate. We can say that, we can argue that this is, we can invalidate. Usually new chemical entity, like structure patents of new chemical entity, we can't invalidate because that is the first time invented for that indication, so you cannot come out. Usually when we say invalidation or non-infringing strategies, it would be around the formulation patents, or API particle size, or morph of the API. Those are the patents which we can challenge, or we can design around these patents. Because the brand companies uses this formulation patents and API morph patents and particle size, these patents, to protect their lifespan in the market. I mean to say that these are the tools which brand company uses to stop the entry of the generics into the market. So these things we can challenge. Then once we make a patent strategy, we go through the patents, and then we will design our strategy, how we have to develop. And then we do API source finalization. Now the first is, there are many things. Sourcing team will come with 5-6 suppliers and says that which one you want. Then we need to identify who is the credible source, and what is the morph of the brand, and who is the how quickly the supplier can supply to us. So based on the timelines, etc., and stability data of the API, everything based, we do some research over this, and then we say we will finalize the API source. Then the next step would be procuring the brand product and reverse engineering of the brand product. Now, since I need to conduct bioequivalence study, now I can't directly jump. I do some formulation, and then I can't go on bioequivalence study, and I can't fail it. Though I said compared to clinical studies, bioequivalence are less costly, but even to do any bioequivalence study, on an average, it would be if I want to conduct in maybe give an example, Jordan, it would be 80k US dollars. Okay, so it's again a costly affair. In Indian rupees, 1 to 1.5 crores. So it is also costly affair. So what we do reverse engineering of the brand. What do I mean by reverse engineering? Let's suppose we understand, like, what is the dissolution profile of the brand. What is the tablet weight, what is the shape, thickness, all these things, physical attributes. Then we will do uh, uh, dissolution profiles, like what is the dissolution profile of the brand product in 0.1 normal HCl, what is in 4.5 acetate buffer, what is the profile in 6.8 buffer. Why we do in gastric, when in a human body, when you take it in stomach, you have acidic media, and then eventually your pH changes. When it goes to duodenum, it would be around 5.5, and then eventually it goes to 6.5 or 6.8. So knowing the physiology of the body, we do in vitro dissolution studies to understand the dissolution profile of the brand. Then we do some then we design our product in the lab, and then we try to compare our product with the brand product, whether my profile is in vitro profile is similar or not. Once we have in vitro profile similar, and then I have stability data, then we will go for pilot bio studies. Before that, we do some other things also. In reverse engineering, we try to identify what is the particle size of the API which brand has used. What do you mean by particle of how? Because if you know, the all active pharmaceutical ingredients can be categorized into 4 classes, like uh, high soluble, high permeable, class 1 molecules, low soluble, high permeable, class 2 molecules, then uh, high soluble, low permeable, class 3, low soluble, low permeable, class 4 molecules. So this way API can be categorized into 4 categories. Now, based on these categories, we need to do, because if it is a poorly soluble, then API particle size is critical to match your dissolution profile. So how to identify API particle size? We will do reverse engineering to understand if the API particle size in a tablet. And also we do some soluble insoluble components. Again, let's suppose if he has used MCC and lactose, I need to understand how much he has used. So as much as this reverse engineering you do, if you deformulate what brand has used, the success for you would be that easy. So we do this reverse engineering of the brand. Also, we understand we do API solubility. Sometimes we develop dissolution method, we conduct compatibility study, and before we start for any product, we do QTPP, means quality target product profile. It is a specification. In other words, in a simple layman language, it is specification or quality which I am targeting, that I will define. Then I will also define CQA. What is the critical quality attributes of the drug product? Let's suppose out of 10-15 attributes which I want to define, what are the critical in this? Like API assay of a tablet is very important, because if my assay of the product is not 100%, let's suppose if it's 70% or 80%, I cannot get my blood profile equivalent to the brand product. So assay is a critical quality attribute. If I say description is not a critical quality attribute, because even the difference between the brand product description, your description change, it doesn't make any difference. Maybe to patient appeal may differ, but then it will not impact your critical quality like safety and efficacy of the product. If you have 140% assay, what does it mean? You are giving extra amount, so it may increase, it may cross your minimum safety level in the blood, so it can give more side effects. So you need to understand what are critical. So based on safety and efficacy, you will define what are CQS of the drug product. Then you need to understand risk assessment and critical material attributes of the drug substance. You need to understand API characteristics. I will show you in the subsequent slides. Then once we do formulation optimization, like dissolution mapping in vitro with the brand, then we have preliminary bio studies. After that, we go for pilot bio studies, like maybe 12 to 15 number of volunteers we will try before we conduct full scale bioequivalency study to understand whether we are matching or not. Let's suppose if we are matching, then we go for a full scale batch. Like the requirement in this regulated countries is minimum you have to take 3 batches with 100,000 batch size. From these batches you have to conduct pivotal studies, because if I am taking 3 batches and my API, I would be using huge API and which are very costly, so I can't just do this without knowing a confidence. So we do this in pilot scale before we go for pivotal submission batches. We take surety, like whatever I'm doing is going to success or not. First I do it in pilot scale. If I get an confidence, then I go for taking the submission batches. From this I will conduct pivotal bio studies, and I will conduct accelerated and real-time stability data with 6 months accelerated and pivotal bio study results and many documents you will file. And once you file this, FDA will scrutinize, and they will give some deficiencies if they believe that they want to understand more about your product. Then once they are convinced, they will give an approval. So the day you file, by the time you get approval, it is minimum 3 to 5 years for a generic product. Nowadays, this has been accelerated. Sometimes you may get within 2 years, you can get an approval. Let us understand what is QTPP. As I said, these are the quality target product profile. Now, what is the dosage form? Like, I need to define it for my product. Let's suppose if brand is tablet, what is the pharmaceutical equivalent criteria? You should have a same tablet dosage form. So you have to define it as tablet dosage form design, immediate release. As again, pharmaceutical equivalent criteria, what does it says? If brand is immediate release, you have to be immediate release. That this way you will define what all the things, what you are aiming at. This is QTPP. From this QTPP, and you have to give, one is target and another is justification behind this. Why you define this in this way? Why pharmacokinetics? We should be equivalent pharmacokinetics or pharmacodynamics in order to be a generic product. Okay, if I cannot have a different pharmacokinetic profile, so that is also generic requirement. So you define what you want before even you start a project based on the literature search. Then you identify from the QTPP. QTPP is 10 to 20 attributes. From the QTPP, you need to identify critical quality attributes which will impact safety and efficacy of your product. These are the subset of QTPP. In CQA, if you see that, you need to identify which is critical quality attribute. In this case, if you see if you in physical attributions, if you go back, these are all the things which we have defined here. From this, we have taken all these physical attributes. In the drug product quality attributes, you see physical attributes. Physical attributes. Does appearance is CQA? No, because it does not impact efficacy and safety. Odor? No, though it can unpleasant taste can have impact on patient compliance, but it will not impact efficacy, safety. No. So like that, you need to put a justification and you have to define which is, you need to identify which is CQA. Assay is S, because assay variability can impact efficacy and safety. How efficacy? If your assay is less than 80%, then it will not show effective. It cannot show efficacy in the patients. If it is higher than 120, then it may have side effects. So that's why it is a critical. Similarly, content uniformity is critical. Dissolution profile is critical. Why? If you are not matching in dissolution profile, it won't get absorbed. If it won't get absorbed, it won't show the efficacy. So that's why it is critical quality attribute. So you have to put all these things. Then what you need to do is you have to do risk assessment of drug substance attributes. Now you identified critical quality attributes. Let's suppose what you have identified: assay, content uniformity, dissolution, degradation products are critical quality attributes. Now you need to think, you have to do a risk assessment. What drug substance attributes are impact these quality attributes? Let us suppose API attributes. What are API attributes? MARF. Can MARF, because MARF can influence assay. Let's suppose when I say MARF, solid state form, it can be crystalline form, it can be amorphous form, it can be crystalline form A, B, C, etc. This can be changed. You don't know what is the brand form is used. So will it impact? Whether whatever MARF you use, assay will not get impact. It cannot impact. Content uniformity. Dissolution, yes, you can have. Let's suppose if brand is amorphous form, if you use a crystalline form, you can have a different crystalline profiles. When you have a different crystalline profile, different dissolution profiles, you can have different efficacy. So that's why it is critical. So drug substance MARF is critical on the dissolution. So you need to identify this. It is a high risk. Then particle size. How particle size of API can impact content uniformity? If you have, let's suppose your tablet weight is 100 milligrams. Your API in that is 1 milligrams. Okay, if your strength is 1 milligram or 0.5 milligrams, then if your particle size is very big, or your particle size is it's not following a bell-shaped distribution, then it can have an impact. Like it may not get distributed properly. So it can have impact. Different tablets can have different contents. So it is a risk. So PSD would be considered as high risk. Then again, if it is a poorly soluble molecule, particle size can be a critical. So that's why it is high. Like that, you have to identify different drug substance attributes which can impact your critical quality attributes of the drug product. Then you have to provide a justification. Like, it is not something like, you put the way you the way you feel it. No, you have to give a justification for each and everything. Then you have to do risk assessment of formulation variables. What do you mean by formulation variables? Apart from API, you use some excipients as well in the formula. Like, you will use bulking agents or excipients like, MCC, lactose, these things. And you will use disintegrants, you will use glidants, you can use lubricants. So these are the formulation variables. You need to understand whether it will have any impact on CQA or not. Then you need to identify it. You have to work out what are the high risk elements. Similarly, if you look at MCC lactose ratio, this can have impact on content uniformity, because if MCC, the grade of MCC, if I use powder grade and my API concentration is very less, it will impact distribution. So that's why I have created it as high. And CCS is high for dissolution, because if this concentration reduces, it can impact disintegration, which will eventually impact dissolution. So this way you have to identify risk elements, and you have to give justification why I am doing all these things. Then whatever the risk elements I am identified, I have to work around these risk elements, because I need to create a robust formulation. So that I need to identify. Then by conducting more trials around this, I need to mitigate the risk. How I mitigate the risk? Let's suppose drug substance, it has impact on dissolution. So how I can mitigate? I need to take a trial with a different particle size. Okay, if my particle size D90 is 10 microns and my release profile is matching with the brand, when my D90 90% distribution of particles are 100 microns, I am very slow in dissolution. So you identified this risk. What is the risk? You first you identify this is high, then you conduct research to understand the risk. Once you understand the risk, put a control strategy. Once you understand, you have to put a specification. Whenever I manufacture batch based on biostudies, my API particle size should not be more than D90 more than 10 microns. This way you are mitigating the risk. First you identify, then conduct experiments, and then mitigate the risk and put a control strategy. Then first you did for API, then you did for formulation variables, then you have to do risk assessment of your manufacturing process. How? Like, let's suppose I have chosen a roller compaction process as a manufacturing process. Now how it can impact, whether content uniformity, whether roller compaction can impact dissolution. Yes, if I compact at high pressure, it can impact my dissolution. So I need to conduct to fix this process parameters. Let's suppose I'm keeping a roller pressure of 10 kilonewtons and I get a very hard, optimum compacts. When I get 40 kilonewtons, I'm getting a hard compacts. So my dissolution profile is slow. So this can impact. So what you need to fix from 5 to 40, when I have conducted 5 kilonewton pressure and 10 kilonewton pressure has given a good compact. So you should put some, I have to put my compaction pressure should not be more than 10 kilonewton. So for each and every unit operation, you have to do this risk assessment, identify risk elements, conduct experiments, and put a control strategy. So once you do all this research, then you have to define your control strategy. Let us suppose API, if these all the things, if you look at on the left hand side of the first column, these are the risk elements. Then you need to see how, next is what was the lab scale data I have used, how I have conducted pilot scale, what was the parameters which you have at commercial scale. Based on that, you are putting a control strategy. Once you put that, you have prepared a development report, and then with pivotal biostudy data, stability batches, submission batches, stability data, and all other modules which I discussed, everything with that, you have to file this in with the US agency. Now with this, you will complete your filing. Then you will get deficiency. If you get a deficiency, you have to address. Once it is satisfied, agency will approve. So this is in brief. Just I try to put it in a brief as as it possible. But then each slide can be a 2 or 3 hours presentation. Again, I try to give a overall picture to you guys. Now we come to the last of the presentation, where what opportunities you have. I mean, where you, after B Farm and M Farm, where you can do it. Let's suppose, in order to enter into the research, a minimum qualification is masters or PhD. And basic research is PhD is must. Let's suppose if you want to go to new chemical entity, you have to do a PhD. And if you want to do into the formulation research, because formulation research is, rather than doing the research in universities, what you do here in industry is for more PhDs. Let's suppose if I say 17 years experience, I have filed at least more than 50 products. Each product, when I filed, I can see considered as one thesis. So in formulations, PhD is not mandatory, but then it is good to have. Then for drug approval means regulatory agency. There would be a regulatory department in the industry, where what you take is, you take the modules I discussed, right? Module 1, 2, 3, 4 data. Different departments prepare different modules. You have to take, you have to compile, and you have to upload as per ECTD format. Again, it is a specialized department every company will have. So you can, after B Farm, even you can do a diploma. Masters is always preferable. After B Farm, you can do a diploma in regulatory affairs. Then you can get an openings in the regulatory department. Then manufacturing, like if you are regular production manufacturing, if you want to enter, you can enter with the B Farm. It is always preferable if you can do MBA, then that would be better. Then marketing, you can enter with B Farm. You can have an MBA as an additional advantage to grow in your career. Then post marketing surveillance, again, as I said, there is a pharmacovigilance. After you launch the product, you have to have a vigilance on your product, how your product is working on the actual patients. So if someone, this is something like if someone has taken your product and they fail any adverse effects, they will complain. So you have to, any market complaints or complaints from the patients, you have to track it, and you have to investigate those things. This is called pharmacovigilance. Even that is also open after B Farm. You can do a specialized diploma for that to get entry. See, again, after B Farm, it's always advisable to go for higher studies like M Farm, MBA. But apart from that, even you can do IIP diploma, like it is like IIT. It is P stands for packaging. There is a university. That university provides diploma in packaging. Packaging is one of the important field in pharmaceutical industry. So if you don't get an opportunity to go for masters, even you can do that diploma, and so you can have there is a separate packaging department in the production, R&D, everywhere. So you can get entry. Also you can do is also you can do intellectual property rights diploma. Okay, so the opportunities you have in industry, you have warehouse, production, technology transfer, packaging, quality control, quality assurance, regulatory affairs, intellectual property rights, pharmacovigilance, marketing, medical representative, government jobs, you have drug inspector, pharmacist, and analyst at central drug laboratory. Even apart from that, you have IT jobs as well, like SaaS. Then because it is a computer software which uses to read your clinical study data. Because for USFDA and all, you have to give SaaS report. And you can go do data mining, business intelligence, which I discussed in my early slide, market intelligence. You can go in that way. And definitely Last but not least, you can become an entrepreneur. You can open an industry, and you can have a medical shop, have wide opportunities. Okay, with this, I end my presentation. If you guys have any questions, most welcome.
Yeah. Yes, Ravindra, thank you so much. It was really a very elaborative lecture. Now I hand over the session to Dr. P. Mary Sulakshana for the conclusion as well as vote of thanks. And we will also have the question and answer session with her herself. Over to Dr. Mary.
Thank you, sir. And thank you, ma'am, for giving me this opportunity. So here is a question, sir. So question from Dr. Deepthi, madam. For pivotal bioequivalence studies, we need reference characterization. Which drug products can be considered as reference products in Indian market, like Orange Book for US market?
Yeah, madam, see, reverse engineering or reference characterization is not a requirement for filing per se, because in order to mitigate the risk, we conduct this one. But in case of in India, we don't have any such list. As I said, our regulate we have not that evolved as of now to understand which is reference product which you need to conduct against bio studies. Usually we will rely, let's suppose we get it from the US, we import these drugs and we will conduct bio studies.
Thank you, sir.
Mary, you can proceed.
Oh, okay, ma'am. Over to Purna, madam.
Mary, it's time for vote of thanks and concluding session. You can proceed, Mary. Over to you.
Okay, ma'am. Thank you. A very good evening to Chairman sir, Principal sir, Chief guest of today's session, teaching faculty, and my dear students and delegates. Myself, P. Mary Sulakshana, working as Associate Professor in Ragu College of Pharmacy. It's my immense pleasure to give a vote of thanks on this occasion. So, on behalf of organizing team, it's my privilege to propose vote of thanks on this occasion. I would first, my heartfelt gratitude to our beloved chairman sir for providing facilities for organizing this event. I would like to thank our beloved principal sir in encouraging and conducting seminars and webinars, both national and international. My heartfelt gratitude and respect to our chief guest speaker, Mr. Raveendra Nagella, for his valuable time and enlightening us with knowledge on formulation development of generic products. Thank you, sir. So initially, you started with a nice story, which grabbed all of us attention to the talk, and also you discussed about various targets, how to identify the targets, and how to combat the targets and all. It's really very interesting to learn such aspects, and also you given a brief steps involved in the flow of various steps involved in the formulation development, and also discussed about various aspects in drug development and approval process, and what are the trials to be conducted for brand and generic products are also discussed, and various differences between the brand and generic products were briefly explained by you. Thank you so much, sir. I would like to thank all the teaching and non-teaching faculty, students, participants for making this event a grand success. Once again, I thank one and all who are present over here. Thank you so much, sir.
Thank you very much. Thank you. It was even nice from it's even it gives me some happiness when I share my knowledge to the budding pharmacist. Even it was my pleasure too.
Thank you, sir.
Yeah, thank you so much,
Definitely.
Ravindra. Here we end the session. You can
Thank
relax.
you, Purna.
And
Thank
seriously,
you,
we didn't expect that you will be taking continuously for this more than 1 hour 45 minutes. It was really
and
a very informative session.
even
Very informative session.
sometimes when I go in that flow, even it was continuous. Thank you.
Yeah.
Okay.
Yeah, thank you. The feedback forms are provided for the participants. Thank you,
Okay.
Ravindra. We will log
Okay.
off now.
Thank you.
Thank you so
Okay.
much.
If there is any feedback, I would be more than happy to listen and know if something more is need to be put in forward in my future presentation. It would be good. Please do share that feedback with me as well.
Sure, sure. Definitely.
Okay.
Definitely.
Thank
We
you.
will be doing. Yeah,
Thank you very
thank
much.
you.
Thank you. Bye.
Anita, you can log off.